And today we're pretty fortunate because we have the experts. We have several guests here from USCG. For now, let our experts introduce them in a bit because they are part of this team as well. Dr. Amy Scaroni is the leader, ring leader of this wonderful group of water women. I don't have to say, most of them are water women, but there are some water men. But they have done a fabulous job of keeping this project together and keeping it going. But Amy is at the Baruch Institute up the coast in Georgetown, and she works with Clemson, and it is the Coastal Ecology and Forest Science Department. Is that right?
Yeah. Yes. The Baruch Institute for Coastal Ecology and Forest Science.
Okay, I'm not going to say that again. And Dr. Stephanie Whitmire is with the same program. And then our main presenter today is a lovely young woman who's working on her master's thesis. So this is a big deal for her today to do her presentation. And she's been probably the chief data aggregator for this, which is a ton of work if you really love data and you know how all that gets put together. So she is the brains behind the data. And then we have Beatrice Calhoun, who also works for Clemson. She's a water resources agent, and she is a Tri-County agent. So I don't know if you remember when we did the water thing, the septic program thing with Tom at Jane Edwards. If you were there, Beatrice was also there to share information. So we're lucky to have her today too. And then Tom Austin is going to talk a little bit about the septic grant program and where we are with that. We've done a lot of good work around here, and there's more to be done, and Tom works tirelessly to continue to secure grants for that. And before we move on, raise your hand if you are a member of my committee, at Outreach Committee, and we've got folks here from that. I think we've got office staff here, and we also have board members. So we appreciate that. So here we go.
Well, we forgot to introduce our water men. We also have Dr. Ty Petty from USC Buford and Tyler, I'm sorry, I forget your last name. Hassig. Yeah. We see each other all the time, but we don't usually email that much. So Tyler Hassig also from USC Buford. So they're part of this larger project team for the research that we're going to talk about today. But I'm actually going to back up a little bit. Tom, here we go. Thank you. Okay. I'm going to back up a little bit. So I'm Dr. Amy Scaroni. I'm an assistant professor at Clemson's Baruch Institute of Coastal Ecology and Forest Science in Georgetown. I do live in Charleston, so I never mind driving over to Edisto. It only takes me about an hour to shoot over here, and been coming a lot over the last couple of years. My involvement in, water quality on Edisto started back really in 2009. So 2009, I got in touch with some folks at the Edisto Island Open Land Trust, Bob, Tom, a few other folks probably in this room, to just talk about the idea of the water quality challenges across the watershed and the possibility of putting together a watershed-based plan, which is somewhat of an official document. It eventually becomes approved by the EPA, and it provides a roadmap to protecting and restoring water quality within a watershed. And so there was a little bit of funding available through DHEC, which is now called Department of Environmental Services, DES, but I always forget, and I'm sure I'm going to slip up and call them DHEC at some point today. They had a little bit of funding available, and so we went ahead, applied for the grant, got the funding, and led an effort to create a watershed-based plan for Edisto Island. So this is available online. I do have only one hard copy. I have a bunch more in my office, so if people want the full copy, I'm happy to probably just drop them off with Tom at some point, and y'all can pick them up across the street at the Land Trust office. But this was an effort of many people. It was actually all water women, on the project team, but we collaborated with a lot of community organizations and local folks, and the challenge was we got funded in May of 2020, so during COVID, the early days of COVID, where we couldn't really get out in the community and talk to people and ask people what their concerns were, what they were seeing around the watershed. So things had to be done kind of virtually, and we didn't get to do as much engagement in person as we had hoped. But I'm just going to give a quick summary of kind of the watershed planning effort and the watershed plan, and then how that led us to the research project we're doing today, or that we're working on, that Brynne will talk about today. So we always talk about the Edisto Island watershed. That's kind of a, I guess, a term we made up. There's actually 3 sub-watersheds that encompass Edisto Island and the town of Edisto Beach. So to do this watershed plan, we had to identify the specific sub-watersheds that we were going to focus on for the plan. So we picked all 3 of these because we wanted to cover all of Edisto Island. We did include Edisto Beach, although for reasons I'll discuss, it is a little bit less of a focus of our recommendations. So, okay, so here you can see that's what the sub-watershed, or what we call the Edisto Island watershed, the outer boundary of those 3 sub-watersheds ends up being in it. Grabs a little bit extra, and there's a little corner of Seabrook over there, but really we were just focused on Edisto Island and kind of the waterways around Edisto Beach. The reason we were focused on it is because South Carolina Department of Environmental Services, formerly DHEC, they have a number of programs that do water quality monitoring, particularly their shellfish management program. They had been monitoring in this area and other areas across the coast going back to at least like 1999. And what they had found over time is that there were a significant number of water quality impairments designated across the watershed. So all the blue dots you see here are for turbidity. So those were locations across the watershed where the turbidity in the water, kind of the cloudiness of the water, was above what they had established as kind of the state standard, where you want it, at that maximum level or below. It was exceeding that. The challenge is kind of to understand is it doesn't have to exceed it every time they sample. It has to exceed it 10% of the samples that they take in a year. So if they are out sampling 12 times a year and 2 of those times the water quality is kind of poor or exceeding the state standard, then they'll actually designate that location or that water body as impaired. So it could be, that 90% of the time things are fine, but 10% of the time, at least 10% of the time, the water quality is impaired, and then it gets put on what they call a 303D list of impaired water bodies across the state. Now, all these other dots are all the non-blue dots up here, and I apologize if anybody has color blindness. Maybe we should think about our color selection a little bit better here, but all these other dots that are really more throughout the tidal creeks, those are impairments for bacteria. And so when I say bacteria, I'm talking about fecal bacteria, so that is bacteria coming from poop. It could be coming from a variety of sources, humans, wildlife, animals, whatnot, but when we see bacteria in the water, that fecal bacteria, that is what the impairments have been designated for. In some cases, they're testing for fecal coliform, sometimes Enterococcus, sometimes E. coli, and Brynne will get into that a little bit more. But you can see there were a significant number of bacteria impairments across the watershed. So at least 10% of samples throughout the monthly sampling throughout the year were exceeding the levels that the state had set as the standard for shellfish harvested waters. So since there are shellfish, oyster beds throughout the watershed, it is considered a shellfish harvest area. A lot of those shellfish beds have been closed to harvest because of the higher bacteria levels. Now, I'm describing this as a sample. If you look at just one of those impaired sites, I just picked one at random, and we had graphed up the DES data going back to about 1999, and we were doing this in 2000, so we were just going up to about 2000. The daily maximum, kind of the state daily maximum concentration here is 43 colonies per 100 milliliters, so that's just kind of the standard unit we would be talking about. You can see it fluctuates, right? So this site was classified as impaired, but all of these sampling dates, the bacteria was fairly low enough that it would, it wouldn't trigger concern if you were only looking at that point in time. But then since they're looking across the whole year, and there are plenty of times where the bacteria levels exceeded that state standard, this site was classified as impaired. And so I think that's the thing we always struggle thinking about the most, because on any given day, it doesn't mean that the bacteria levels are high, but overall, it means that they are high enough that kind of out of an abundance of caution, DES would classify this site as impaired, and sometimes that could lead to restrictions on shellfish harvesting at that location. The turbidity is the same concept. It's a little bit different because the turbidity data for the watershed, most of the sites had only been monitored monthly for a year, so we didn't have this 30+ year record. Over a year, you're taking 12 samples a year. If 2 of those exceed the state standard, then they're going to classify it as impaired for turbidity. So in this case, 1, 2, 3, 4, 5, 6, I guess, exceeded out of 1, 2, 3, 4, 5, 6, 7. So, like 6 out of 13, that's more than 10%, so it's classified as impaired. But that was those turbidity sites, which are mostly in the Edisto River, haven't been monitored as regularly. So it's not continuous monitoring. So that was something we wanted to keep in mind to see, were we still seeing turbidity issues now if we went out to sample.
Excuse me,
Sure.
what causes turbidity?
Can
You
I
get that?
pass that off to
Okay,
Brynne?
sure.
And if you don't feel like your any questions answered,
Okay.
ask it again and we'll get to there.
Okay.
But she's going to dig into the details
Okay.
of the turbidity and the bacteria. So when we put together this watershed plan with a lot of community assistance, the goal that, as we saw it, was we really wanted, our vision or our goal for the watershed was to reduce the number of bacteria and turbidity impairments across the watershed, so that you would be removed from that list designating the 303D list, designating impaired water bodies, and eventually reopen a lot of the shellfish beds across the watershed to harvest. So we see that as the big overarching goal. It sounded very daunting to us at the time. This was the first time any of us had been involved in putting together a watershed plan. But we looked around the coast and there's other watersheds that have done this successfully. Merle's Inlet had a bacteria impairment. They made a watershed plan, they made a concerted effort, and they've actually been able to remove that. I think the May River and some of the work around the town of Bluffton, they've done watershed plans and they've been able to remove some of their bacteria impairments. So while it seems like a daunting task, there are success stories, and we do think eventually are hopeful that similar successes can be seen in the Edisto watershed. Just being realistic that it can take some time. When I started working through this, it was 2020. We're already here 5, almost 6 years later. And it's still a work in progress, but it's work that we're continuing to do, and hopefully we'll expand. And there's now a lot more folks getting interested and kind of looking to Edisto as a place where they might want to help with their research efforts as well. So if we kind of think about it as a long game, we are hopeful that we are making progress towards. And when I say we, I don't mean me, I mean everybody, local organizations. Tom will get to talk later. There's a lot of other folks kind of in the community doing things now. So if you're wondering what a watershed plan is, and I gotta wrap this up because I'm probably taking too much of Brynne's time. It is this kind of EPA has a requirements about how to put together a watershed plan. The key things are that you want to identify all the potential pollution sources across the watershed, identify management measures that can address those pollution sources. Then you even have to go do some modeling and estimate what the pollutant load reductions would be if you took some management measures. And then there's a whole bunch about, recommend sources of funding, recommend outreach. That's a little more of the procedural part of it. But the great news is when you get when you submit your watershed plan, the EPA approves it. It opens up eligibility to some sources of federal funding that you wouldn't be eligible for otherwise. So because the watershed plan was approved by the EPA, the Edisto projects within the Edisto Island watershed are eligible for federal funding sources, such as, Tom will talk about the septic improvement program. They were able to get some EPA 319 grant funding that is only eligible to watersheds that have a watershed plan. So just getting the plan complete in and of itself was a great step forward to being able to do a little bit more to work towards some solutions. So when we looked around the Edisto watershed to identify potential sources and solutions, we were looking, specifically at sources of bacteria and specifically at sources of turbidity. Now, we really looked at all potential sources that were probable within the watershed. So that was, driving around, talking to people, looking at a lot of GIS maps, trying to figure out, do we have hogs on the island? Are there deer? Are there raccoons? Are there septic systems? Are there wastewater treatment plants? So all the places we know bacteria can come from, looking at all that and trying to kind of whittle it down and rule things out and figure out what makes the most sense for Edisto. We did the same thing with turbidity. We did a lot of geospatial analysis, so GIS maps. And when I say we, I didn't do any of that because I am not too tech savvy, but we had some GIS folks that were able to map all different things about the watershed that gave us a really good picture of where, maybe the soil is poorly drained and septic systems in that location might be more likely to fail, or areas that are more likely to, flood during high tide, or, all sorts of stuff. This is just a snapshot. If you look at the watershed plan, there's dozens and dozens of maps in there. We also tried to get some feedback from the community, so some people did submit online concerns that they had throughout the watershed, and we had some key stakeholders representing groups on the island give us feedback once we're finally able to meet in person. And what we really came up with as the most probable sources of bacteria were going to be failing septic systems, discharges of untreated wastewater from boats. That wasn't even something we had thought of, but that's something we heard from the community, that they knew there were people that would just empty their boat out into the water. Waste from wildlife, livestock, and dogs, and perhaps stormwater discharges from detention ponds, which would probably apply more to Edisto Beach. And then turbidity, somebody asked about kind of sources of that. A lot of that can just be stream bank erosion. If you have erosion, that soil and sediment is going to end up somewhere. If you have kind of bare land, dirt roads, you can have erosion from there. Runoff and excess nutrients. And Brynne will talk a little bit about the kind of nutrients versus sediment when she presents. So, and I'm not actually going to go through these, but what we did is we made a bunch of key recommendations about what we could do within the watershed to address each of those potential sources, and then kind of how much pollution reduction we could get from those. So we had residential best management practice recommendations, we had ones for agriculture, we had ones for kind of within water bodies, things like no wake zones and living shorelines. We had things for wildlife, because we do know the wildlife go to the bathroom, and that is natural. There are natural sources of bacteria, but sometimes when you have more concentrated populations, you can end up with higher levels, and so are there ways that you can manage things on land to reduce maybe congregation of Canada geese at the edge of a stormwater pond, for example. We had some forestry ones just for folks that may manage their land for timber. And then we went through very specifically and prioritized areas within the watershed where each of these might be most effective. And so that's all in. I mean, the watershed plan is like 120 pages long, so there's a lot of that in there that you're welcome to look through. And then we wanted to think about, with limited resources, how do you get started? We, really want to focus up here where everything's high impact. We're going to have high impact on reducing pollution. But what about these ones that may be low impact, but it's low effort? So, there's some low-hanging fruit things that people can do that could still have a difference. And so that's a lot of kind of what we've talked about, but we've seen moving forward that things are starting to happen. Tom's going to give an update on the septic improvement program. That was kind of a spin-off of the watershed plan. There are some living shoreline projects that are now planned within the watershed to address some erosion from stream banks. That's just a random photo. That's not one of the locations that I was referring to for a living shoreline. We know that a pump out station went in at the Edisto Marina, so that can be a location where people can empty their waste so that it doesn't go directly into the water. And then if you're interested in finding out more, I can try and get a hard copy into your hand, but you can also download the PDF of the plan online at edistowatershedplan.org. And then that brings us to the next part of our talk, which is we finished all this without being able to collect any data of our own. That was, our funding said you can't collect any novel data, you just have to look at what already exists and try and make sense of it. But what we're finding is like, well, we need more data. We don't have enough information. So we, Ty, Stephanie and myself, Tyler, Brynne, Tom, we got grant funding from South Carolina Sea Grant Consortium to do a little more water quality monitoring and see if we can make sense a little bit more specifically about where the bacteria is coming from and where the turbidity is coming from. Turbidity. And so that's what Brynne is going to talk about next.
All right, hello everyone. Thank you so much for being here. Again, my name is Brynne and I'm the master's student on this project. And so now we're going to take a deep dive into the water quality monitoring project that we've been doing for about the last year and a half. To really try and tackle some of these data gaps that the watershed-based plan kind of opened our eyes to. So our main objectives of this project are to narrow down and determine the source of fecal pollution and turbidity across the Edisto Island watersheds. So again, we kind of already talked about our project study area, but we are looking in the South Edisto River, which is the light blue part here. And this is where we see a lot of those turbidity impairments that Amy was talking about. We're also looking at this medium blue portion, which is the Belva River, North Edisto River. And in this upper portion here, near Highway 174, is where we see quite a few bacteria and turbidity impairments as well. And then thirdly, we're looking at this darker blue portion in the Store Creek, and we see quite a few bacteria impairments here as well. So next, I'll go a little bit about some of the sources of fecal pollution that we're seeing in the watersheds. And again, we're talking about how poop can end up in our waterways. So in rural environments such as Edisto and in coastal areas, septic systems are one of the most common ways of residents' wastewater disposal. And these are great systems, especially when connection to sewer is not available. However, if these systems are failing, if they're not properly maintained, they can lead to fecal contamination in the surface water. Another thing is, as Amy mentioned earlier, the improper disposal of boat waste can also lead to fecal contamination. And a good example of mitigating this has been through the pump out station that was installed near the Edisto Marina in the Bay Creek area. And a nearby impairment was recently taken off of that 303D list after this pump out station was installed. And so we know that these are great tools to use in order to limit fecal contamination in rivers and tidal creeks. We're also looking at dogs and pet waste, as if pet waste isn't picked up, it can, if it rains near a yard and a waterway, that can end up into the waters. We're also looking at birds, livestock such as cattle, horses, and then we're also looking at wildlife such as deer. So why do we care about managing for fecal bacteria? Well, in coastal South Carolina, shellfish harvesting is one of the most important recreational and economic activities that the waterways offer. And so South Carolina DES does a great job of monitoring for shellfish harvesting. And so they're using a type of bacteria called fecal coliform. Can you see? I'm sorry. Here, let me move out of the way. Okay. So for fecal coliform standards, they are looking at, again, that monthly average for an impairment site cannot exceed 14, a concentration of 14, and then the daily maximum cannot exceed a concentration of 43. And so if this happens 10% of the time, as Amy mentioned, they'll deem a site as impaired for fecal bacteria. So annually, the South Carolina DES, they are, based on their water quality monitoring efforts, they will release a shellfish harvest classification for the entirety of South Carolina, but this is our Edisto Island one, and this is from 2024. So, or 2025. It's the most recent one that was available. And so these green areas up here, these are approved for harvest, meaning that the water quality is good in these areas and shellfish can be directly consumed and harvested. Conditionally approved is in that beige color, and it's not shown on this map. I know it looks kind of beige up there, but that's not part of the shellfish harvest classification. And so if this was on the map, this would be, they're normally open, but if it were to rain or if discharge from, say, the Edisto River was super high after rainfall upstream, they might close these for harvest. And then the orange portion, these are restricted for harvest unless you have a special permit. And then the prohibited is in brown, and these are often adjacent to like a marina or a wastewater treatment facility. And so these are closed for direct harvest and consumption. Okay.
I have a question.
Yes.
Store Creek, there's all in gold, is restricted unless they have a special permit. There's a lot of commercial crab lots in Store Creek. Are crabs not affected by this, or is that, I mean, I see oysters and.
Yeah, that's a great question. I don't know if I know the answer, but I think Amy might. Perhaps.
We do, so the regulations are different for commercial versus recreational. I do know that because commercial, they take or at least for shellfish. Crabs, normally we have a shellfish and a food safety person that we like to bring with us to presentations because they're usually better at answering kind of the living marine resources aspect, but I believe crabs, it's not, because the oysters are like the sessile organisms that are like filter feeding, and I don't know if you want to add in, but I don't think the restriction is on crabs because they are not like bioaccumulating bacteria. My colleagues are not in. Okay. But we can also look into that more specifically. I do have some fact sheets I meant to bring that some of our colleagues have made on those topics. We can make sure to have those maybe shared or emailed around or something. That is a good question.
Yes.
Why would special permits be permitted in a restricted area? If
Yes.
it's restricted,
Yes.
it's not good, right?
Yes, so like Amy mentioned, if it's for maybe a commercial, they will take these oysters and then they can wash them, clean them, do whatever they need to remove whatever has been accumulated. I believe that.
I think they can incubate them in other waters in the estuary that are considered clean. And yeah, it's like a week or I forget that's the time period, but then that is assuming to flush any of the filtration from the bad water to the good water as you clean them out. Yeah, it could be. Yeah, that sounds right.
Yeah, so I'm going to clarify for everyone. So I meant to say this before, but shellfish, they're called filter feeders, and so they stay in one spot wherever they grow, and then the water column just moves through them, and so they take any of the pollutants that the water has, and then they can accumulate these pollutants. So that could also be toxins, as well as pathogens from bacteria. And so that's why we're worried about bacteria in shellfish, is because they don't really have a mechanism to release that bacteria, it all just gets accumulated. So another way that South Carolina has monitored for fecal bacteria is through recreational monitoring. And so when we're talking about the bacteria Enterococcus, they thrive in saltwater environments. And so that's why we're focused on Enterococcus in South Carolina's coastal communities. However, if we were more in the upstate near lakes, we might focus on a bacteria called E. coli because they thrive in freshwater. So the Enterococcus state standard for recreation is a concentration of 35 or a concentration of 104 for a daily maximum. And so if any of these are exceeded, then 10% of the time, again, South Carolina will consider that an impaired site for recreational. So contact recreation and pathogenic bacteria exposure can be problematic for human health because it can increase the risk of gastrointestinal illness via if you ingest the water accidentally when you jump in, or if you maybe have a cut on your leg and you jump in and the bacteria can enter that way. So all of these are human health risks associated with recreational bacteria monitoring. So next I'll talk about turbidity and some of those potential sources a little bit more in-depthly. So sediment erosion is one particular concern for turbidity in environments. And again, let me clarify what turbidity is again. So it's kind of the cloudiness of water. So if you think of a super highly turbid area, that would be some really cloudy water, maybe it's murky, probably can't see to the bottom. But if we're talking about a low turbidity environment, that would be super clear water. You might even be able to see to the bottom if it's shallow enough. So when we have sediment erosion, this can come from maybe the land. This can be pretty common in construction sites or around roadways where maybe dust and particles from whatever was occurring on the road. After it rains, this rainfall will potentially go into a drainage ditch, and then that can end up in the waterways as sediment erosion. Shoreline erosion is another, oh yeah, sorry.
My question is about the turbidity and the measuring of it. Certain times of the year, the water, and we know, is gin clear.
Mm-hmm.
You know, in the wintertime, that kind of thing.
Yes.
And so how do we determine different times how it differs between, you know, month to month? And then we also said that we only tested once a month for that turbidity. So where are we on that?
Yeah, so I'm kind of giving it a big overview, but we are going to talk about that in just a couple of minutes. I'm going to show you some of our data that we found. But you're right, it does fluctuate kind of throughout the year depending on conditions, depending on wind, rainfall, tides, all of that. So I'll dive in a little bit deeper in a little bit, if that's okay. So another way that we can see turbidity in environments is through shoreline erosion. And so this can occur from wave action or even like boat wake from hitting up against the edge of the marsh constantly, and then some of that marsh material can end up into the water and suspended into the water column. And then sediment resuspension. So as we've mentioned, coastal environments are super dynamic systems. So wave action and tidal action is constantly at play. Water is always moving. And so turbidity is just kind of a natural part of coastal systems because sediment is constantly being churned up due to waves and tides. The third thing that we're considering is algae, and algae are plants in water. They can grow too much, which can be not such a great thing, but plant life in water is natural, so it's all about finding the balance of how much plants can create a healthy ecosystem. But when we have too many nutrients, such as from fertilizers, it can lead to something called an algal bloom. So when we have fertilizer, manure, or a leaky septic system, this can enter waterways through runoff. Again, like I've mentioned, runoff comes up a lot in water quality. So then we can have some excessive algal growth or too much plant growth. And then it can start to look like this, which doesn't look great. And so if we start seeing all of this plant biomass, it can use up available oxygen for the other organisms that are living in the water. And it can create toxic conditions for both fish, wildlife, and then the humans who also are using and recreating in this water. So when talking about managing turbidity, South Carolina is also doing a great job of monitoring for turbidity. So as you can see, I mentioned earlier, a lot of the impaired sites for turbidity are in that South Edisto River. And so when a site is deemed as impaired, it is exceeding 25 NTU, which is just a generic unit for turbidity. Some of the consequences of turbidity, it can be a recreational nuisance, meaning it's not the most pretty water to look at. Personally, I don't really want to swim if I can't see like where my feet are. And then it can also visually impair wildlife. So if a fish can't see where they're going, that can probably lead to some issues. It can also reduce plant growth. So if plants outside, they need light to grow. Same thing in the water. If light isn't able to reach them, then they can't grow. And like I was talking about earlier, plants are important to ecosystems. Too much plant growth can create those toxic conditions we were talking about, but we want just enough to keep the ecosystem healthy. And then also, fecal pollution can be exacerbated or increased with increased turbidity because bacteria can attach to turbidity particles such as sediment or just other materials in the water, and then they can accumulate and grow, and then that can increase fecal pollution as well. So where have we been looking? So based on what we know from South Carolina DES's monitoring efforts, we've chosen 19 sample sites to look at throughout our 15-month sample period. So we started in June of 2024, and we just concluded in August of this past year. So we're looking at some of these old sites, which are in the South Edisto River, and we're only looking at these for turbidity on a monthly basis. We're also looking within the tidal creeks, and so all of the tidal creek sites we're looking at both turbidity and bacteria because that's where we see some of those impairments. And so the blue sites we're looking at those monthly, and then the quarterly sites are purple. And we added in our 6 additional quarterly sites to kind of get some seasonal variation to see what's happening on more of a seasonal basis. And then I wanted to give a special shout out to Bob Hare because he was a volunteer for us who was able to go out once a month and collect some additional turbidity samples where we weren't able to during our normal sampling trips. And so that has been super helpful for our data analysis. And yeah, we're just really grateful for that. So when we're in the field, sorry, this chart might look a little confusing, but I'm going to go through it. So we have our handheld sonde, which is called a Y-side, and we'll stick it in the water, and then it can give us a bunch of different water quality parameters like temperature, pH, dissolved oxygen, things of that nature. And then we're also taking water samples back to the lab to analyze them. And so I'm going to start on this row, and then I'll move down here. But so for our turbidity analysis, we're looking at total suspended solids. And so that's when we filter the water and we look at everything that was floating in the water. So that might be sediment, that could be plants and algae, anything if it's in the water, we're measuring it. We're also looking at something called chlorophyll A, and that is an indicator of algae growth and presence in the water. And then we're also looking at the nutrients nitrogen and phosphorus. And these are great indicators of plant growth in water and how well plants can grow. For the bacteria side of things, we are looking at host-specific qPCR, which this is how we're trying to figure out what our source is. And so we'll take our water sample and extract the DNA from the fecal bacteria in the water, and then we'll look for matches based on the known sources that we're looking for. So that would be a human, dog, avian, or bird, things of that nature. And so then if we get a match, then the qPCR and a reaction will inhibit, or will amplify, and then we'll know that we've gotten, that we have the source in that bacteria. And then we can also see the concentration. And then finally, we're looking at the concentration of Enterococcus E. coli in our water samples. That's similar to what South Carolina DES is doing for their impairment thresholds. So now, hopefully, I'll answer your question with this slide. So for our turbidity by month, again, we are only looking once a month, but we try to sample during different weather conditions and tidal conditions to kind of capture what's going on broadly in the watershed over the year to year. So this plot right here, on the x-axis we have the date that we sample, and then on the y-axis we're looking at turbidity. So the red dots are when we exceeded that turbidity threshold from South Carolina DES, and then the blue dots are when it's below that threshold. I will note, so a lot of these blue dots are clumped together, so it looks like we have more red than we do, but 37% of our turbidity samples were greater than 25. And As you can see, this December date was really high, and this was after a few consecutive days of higher than average tides. So that is definitely something to consider with turbidity, as well as like rainfall and wind action as well. So based on our findings so far, turbidity is primarily driven by sediment across Edisto. This map right here is showing a pie chart for each of our sample sites. And so the darker portion is our sediment portion of turbidity, and then the green or mint part is the organic or the biological plant side. And so as you can see, all of these sites are mostly sediment, and so we can say that sediment is primarily driving the turbidity that we are seeing on Edisto. So what can we do about that? Well, sediment and erosion control are really great ways to keep sediment from entering into the watershed. So this can be done through some of those best management practices that Amy said, and then we can also talk about more at the end. But really trying to find ways to reduce runoff from land into water. And then also super important is adhering to those no-wake zones to reduce the amount of boat wake that's leading to that shoreline erosion from the water hitting the marsh's edge. So what about algae? I talked a lot about sediment, but is algae at play? And it is. So while sediment is driving turbidity, algae is still present in the water. Algae do naturally bloom, kind of like plants outside. Their natural bloom cycle is in the spring and into the summer, and then it can last through the fall. So this plot I have right here, the x-axis is the season that we sampled, and then on the y-axis we're looking at the average chlorophyll A. And we see summer is much higher than all the other months, and then spring is also a little bit higher than the others as well. So the summer chlorophyll A average actually exceeds the poor threshold that a group of water quality professionals in South Carolina kind of distinguished for coastal South Carolina. And then the spring average exceeds the fair threshold. And then when we're looking at chlorophyll A community, we're seeing mostly diatoms, which is a great thing because diatoms are super important for a healthy ecosystem, and they're not really indicative of like one of those harmful algal blooms that I was talking about earlier. So when we are talking about harmful algal blooms, the main community member that we would be concerned about is like blue-green algae. And as you can see, it's pretty low for most of those months. A little bit proportionally higher in the summer, but not anything crazy to note, which is a good thing. So even though algae aren't maybe driving turbidity, it's still really important to be diligent about limiting fertilizer use and reducing runoff from the land as well. Because especially in these summer months, spring and summer, when your gardens are in full bloom, or you're trying to get them into full bloom, it's important to only use the amount of fertilizer that you need. Okay. So for some of the bacteria results, next we have a similar plot to our turbidity one, where we have month on the x-axis and then Enterococcus on the y-axis. And again, the red dots are indicating when we exceeded that impairment threshold of 104, and then the blue dots are when it was below. Now, as you can see, only 6 instances did exceed that Enterococcus threshold. So we didn't see it many times, but it's still there. And then whenever, or in March, it's not really shown here, but this month right here, March, it was directly after a rainfall event, so that could be why we're seeing some of those higher values there. So for our qPCR findings so far, our analysis is still in progress, but so far we don't have any quantifiable like matches that we found a source. So I will say qPCR is a super duper sensitive process, we're working with really small volumes, and so since we've seen so many of these sites below that impairment threshold, that could be a reason why we're not seeing any sources. So just because we're not seeing a source doesn't necessarily mean that bacteria is not present. It just means that we're not seeing enough bacteria to get a detection. And as I mentioned earlier, septics are very widespread throughout Edisto, and so we can't say that septics aren't causing the problem. We just don't know yet. And then one more thing to note is that a lot of the docks that we sampled off of were often covered in bird poop, and so we're working to increase some of our markers, so we're working to find more. So we use a marker in order to run the qPCR, and so we're trying to add some more to our library and expand our bird marker to capture gull, which could be contributing to the bird poop. So in conclusion, sediment dynamics are largely driving those turbidity conditions across Edisto. And so some of the things that can be done include no wake zone adherences, so we're really making sure to follow those. And then some other things such as reducing erosion. In the Big Bay Creek, there's a living shoreline, which is a big oyster structure that's really helping to stabilize that marsh's edge and reduce erosion into the marsh that way. And then while algae still does contribute, it does more so in the spring, fall, and summer, so it's important to fertilize responsibly. And so one of our biggest recommendations is to get a soil test with Clemson Extension, and they can help determine what fertilizer would be best for your soil and how much to use. It's also important to only use the minimum amount to be effective, only applying when it's dry, so not before or after a rainfall, and then using slow release nitrogen and phosphorus, and then not applying fertilizer close to a water body or an impervious surface like a roadway or a sidewalk. And then in conclusion for our bacteria, our project is only a small snapshot of what's going on Edisto. And so just because we did sample on generally good water quality days, it doesn't mean that water quality on Edisto is good every single day. And so it's really important to be diligent about how to reduce the amount of pollution or fecal contamination that is entering into the watersheds. And one really great way is septic tank maintenance, which should be pumped out every 3 to 5 years, and just general good practices surrounding septic tanks. It's also important to pick up pet waste, especially in public spaces like along the beach or close to waterways. And we do know that rainfall events and king tides typically are where we see the biggest spikes of fecal contamination. And so reducing contact recreation directly after rainfall events is a really great way to reduce your risk of being exposed to pathogenic bacteria. And with that, I think I have some time for questions, hopefully. And then I wanted to acknowledge our team, all of our partners, and especially those of you who are here who helped us with our sampling process, Bob Hare, and everyone else who allowed us to use our docs. We really appreciate your help, and we could not have done it without you. Yes.
Question?
Yes.
It's about sampling.
Okay, yes.
When you do your water samples, are you doing them at the same tide level
Yes.
every time?
Yes, we did it at the ebb tide every single time. The idea of that was really for bacteria to capture what was coming from the land on the way out instead of what was flooding in the ocean. Yes.
But on the sampling thing, these are all snapshots you're taking a look at. Is there technology out there that can continuously monitor that so we can get a better idea of what's really happening?
Yeah, there's a lot of really great technology out there. They are very expensive and outside of the budget. But I think people are interested in that on Edisto, and so I think they have
Yeah, I will add that I'm actually working on a grant proposal with some colleagues, so I can't say too much about it. But it is looking at the idea of maybe creating sensors for real-time bacteria monitoring, and we're hoping that if we get funded, we would use Edisto as our case study location to test them out. So the commercial technology now would be very expensive, and at least at our kind of level, cost prohibitive, but we're trying to come up with some innovative ways to maybe do things on a lower budget.
I have a question.
Yes.
How close does a septic tank have to be to the water system in order for it to leach into it?
I'm not sure if it has to be a specific distance.
Yeah, I'll let you chime in also, but the concern is more so the distance between the groundwater and the bottom of the septic tank drain field, because the groundwater can be a strong conduit if you're not getting full treatment in the drain field before it reaches the groundwater, and then the groundwater will move out, to the tidal creeks. If you are right at the edge of your property next to the marsh, and your backyard is flooding during high tides, then obviously that connection is direct and apparent. But just because you're close to the water, if you are maybe at a higher elevation, you have good separation distance with the groundwater, and your tank is well maintained and your soil is not poorly drained, it could be fine. And I think that's the hard thing about septic is it is very site specific, and it's also very dependent on how you're treating it. So
Yeah, all the things you were saying is that the biggest thing is your elevation. If you don't have enough room for your drain field to treat your water, then that's not a safe location for that drain field to be in general. And that could be upper or close to the water edge. If you don't have that space for it to do its job, that's not good.
And B did bring some resources with her, so I think we still need to save some time for Tom, but before people leave today, please swing by this table. B has some informational materials and would be happy to chat more specifically about septic maintenance, because that's something she talks about a lot throughout communities.
This isn't a question as much as a comment. I think the way you get people invested in this project, this change, is by emphasizing their inconvenience. People get really upset when a beach is closed because of high bacteria, or if they can't safely go and collect their oysters. I think we just have to make people connect the dots.
I agree.
Help them with that, in terms of education, because that's really the only thing that will motivate most people, is how it affects them.
That's a great point. Yeah.
One more question. You brought
Yes.
up technology, and I was wondering is there any movements toward being able to determine what the source of the fecal matter is, whether it's bird, dog, raccoon, human?
Yeah.
Is anybody doing that? And can it?
Yes, so that's what we are doing with our qPCR. So it's just that we haven't seen any of those matches. So the point of this project was to do that, but when the bacteria levels aren't high enough, or like if the concentration of bacteria is just not, like, sometimes it needs to be 12,000, but we're only seeing 100.
Right.
And so we're not always seeing high bacteria, meaning we're not always able to get one of those matches.
Yeah. The reason why I bring that up is because we have one of the largest bird rookeries on the East Coast,
Yes.
just
Yeah.
off our beach.
Yes.
So, yeah.
And so that's one of the reasons why we're looking at that bird marker. And we're hoping to expand that bird marker and improve it in order to maybe get some matches that we weren't seeing before. But our analysis is still in progress, and so we're hoping to have some better answers. I know that might not have been the answer that you guys all came here to hear, but we are working on still improving our methods.
And just to add to that, so Brynne, the project is still underway, and because, and Tom always reminds me of this, because we're always so disappointed when we find low bacteria levels, because we want to do the microbial source tracking and find the match, and Tom's like, well, keep in mind it is good for the watershed that we are regularly finding low bacteria levels. So one of the things that Brynne wrapped up her monthly sampling for 15 months in August, but our partners, our watermen, as we're now going to call you, have continued to go out and have actually we've targeted some locations that we thought they weren't necessarily coinciding with where the impairments were on the map, which is how we chose sites initially. But we tried to brainstorm, like, where are we just going to see really high bacteria? So we looked at some stormwater outfalls, and we looked at, I think, a pond. We took a sample from the beach. So we tried to go to places where it's like, even if it's not the areas we were concerned about, where can we just go find high bacteria, and then try this QCPR microbial source tracking to see if we can get some matches there to at least show, you know, like what some of the sources are out there, even if they aren't the ones that we were the locations we were most concerned about. And so that is still a work in progress. So kind of stay tuned. You know, Tom, when he asked us to come talk, we were like, well, we're not quite finished. And he's like, that's okay. Like, you can always come back. So it is a work in progress, but we're really excited to hear the questions that you all ask, because that helps us kind of stay focused in the right direction as we kind of continue our investigation.
Question.
Yes.
And are there any like rules of thumb for the turbidity measuring? Like if you went out to your dock with a glass measuring dish or a bottle and you held it up to a piece of paper with some writing on it and you could read it or not read, can you like guesstimate your turbidity level?
That's a really good question.
They actually have, you can make your own, they call it a Secchi disc.
Yeah.
And it's just a disc, and it's like white and black, and like a quadrant, like white, black, white, black. And you can, you know, increment your rope, and then you can, however far down, right?
Oh.
It gives you, it's a transparency, right? So turbidity is what's floating. So how easily can you see that? And if you can get it, you know, and you can measure down, then you can measure potentially off of your dock how that's changing. Monthly, weekly, after rain, whatever you might want to do, and that would give you an idea on your own personal, in that own personal little creek area where you are, what your turbidity measurement
You poke
is.
it down in there and it's
And I could actually probably send Tom some instructions
Well,
on.
if you volunteer for the South Carolina DES Saltwater Adopt a Stream program and become certified, the Land Trust has a rental water quality kit that you can borrow that has a Secchi disc in it.
Yeah.
And also a transparency tube, which works much better because our currents are usually too fast for the Secchi disc.
So,
But
And we will offer Adopt a Stream training again. It is almost an all-day training, and we generally do it at the State Park dock. But we'll offer that again in the spring, and we are always looking for people who have docks and access. Some people have started it. You have to keep up with it. It's hard to keep up with. I'm guilty.
me too.
So, at any rate, we will offer Adopt a Stream training again at some point in the spring when they can come to us. So if you're really interested, it's a great time to contribute to their data. I think it all eventually kind of meshes together.
And that's kind of a statewide program, so they look at it for freshwater and saltwater across the state.
Doesn't
But
do
you can
bacteria, but
buy-
it does everything else.
You can buy a Secchi disc pretty inexpensively. I've bought cheap one before for maybe like $10 or $12. And it again, you just lower it down in the water, you wait till it disappears, you pull it back up, you wait till it appears, and you kind of measure the depth. And so that's something that you could do regularly, kind of establish a baseline, and you would see when it changed. So then you would know when it is much more turbid than usual, and then less turbid.
The key is marking the line.
Yeah, make sure you mark
Mark
that
the
line.
line.
That
would be
rope
your
that you're,
measurement.
yeah, the rope you're lowering it up and down on, make sure it's
So you're not having
There's lots of YouTube videos about it too, so if you wanted a DIY one, or email us, email the,
Yeah,
we can make you a video of how to do
do a
it.
test run together.
General rule of thumb is if you can't see the bottom of a 5-gallon bucket, it's probably pretty bad.
Okay, there we are.
All right, so I don't have a particularly long presentation for y'all, since Amy and Brynne did a fantastic job covering most of my PowerPoint. So really, kind of thing I want to highlight is just, in Estawan, looking at it as a whole, we are about 50% protected, looking at the island. We're part of the Ace Basin, which is renowned for its water quality and its land protection, and we're also in outstanding resource waters. Yet at the same time, we still have water quality issues here on the island. These can have impacts to the wildlife and the ecosystems that us as a land trust are working to try and protect, restore, and enhance. But then there's also very significant human health risks. And then at the same time, that comes with a loss of access to our natural resources through loss of ability to do traditional cultural things like harvest your own oysters out of the creek by your house for an oyster roast, that kind of stuff. There's a lot of knock-on consequences that come with poor water quality that we don't really think about until we start looking at the big picture as a whole. So we started trying to come to grips with this back in about 2019. And with that, we got connected with Amy, we worked on the watershed-based plan, and so this gave us kind of a really good picture of kind of the state of water quality on Edisto Island and how we could go about looking in at this and trying to address the water quality issues that we have. And from that, the two big things that we found out, like we just heard, is that we have an issue with excessive turbidity, mainly in the South Edisto, but then bacteria pollution is the largest water quality issue that we have on the island. 70% of our tidal creeks are impaired with fecal coliform bacteria. And as a consequence of this, a lot of our shellfish beds are restricted or otherwise prohibited for harvest. And if you look, that's basically every creek that's next to where a lot of people live is closed, which means that the folks who have lived there for generations can no longer access oysters unless they take a boat to a boat landing, get it, go all the way out into the South Edisto River, or way out here onto the North Edisto River and along the Bohicket and all of that over here. So it really has a lot of consequences, large scale, for folks who have relied on oyster harvesting here on the island. Additionally, there's just elevated health risks for anyone who recreates in the water, anyone who fishes. Just think about every time you put the corner of a cast net in your mouth when you're throwing water, you're potentially ingesting pathogenic bacteria that are floating in that water column. There is just a very broad level of low-grade increases in human health risks that come from poor water quality, specifically with fecal coliform bacteria. As Amy highlighted, water quality issues are not bad all the time, but it's not necessarily obvious when they are bad. Water really doesn't look any different. And so these are really surreptitious risks that are out there in the whole watershed that people are getting exposed to, and they don't necessarily know whether or not there's no bacteria in the water today or if it's at astronomically high levels. And so these poor water quality has these sort of low-grade, long-term effects for everyone that uses that waterway. It's also what results in swim advisories and beach closures when levels get particularly high, especially in the summer months. With high turbidity, the main impacts is that it can really mess with the ecosystem. It can increase algal growth, increase algal blooms. It can also impair how certain wildlife hunt. It can degrade habitat for certain species of aquatic life while also favoring others, and that can cause imbalances in the ecosystem that can have long-term impacts through trophic cascades. And it's really not something that can be easily predicted, and so it's something we just generally want to get a handle on before the presenter just randomly shuts off on me. Crisis bird. So, but poor turbidity is also a sign of a lot of erosion in our waterways, which is not a good sign, because if we have a lot of erosion, that means we're losing oyster reefs, we're losing marsh, we're potentially losing upland, which is not good for habitat conservation, and it again has all those knock-on effects, and additionally increases suspended sediment, increases the suspended nutrients, which can then have those further trophic cascade effects. So, what's the cause of this? We still don't know, but we're working on it. There's no big pipe we can just point out and say, there's the issue, get an EPA. This is clearly and obviously some sort of large-scale chronic effect caused by a lot of small sources all across the watershed. If we look at it, going back to this, if you look over here at some of the more undeveloped creeks along the North Edisto River, you really don't see any water quality issues, and these are the places where there's minimal development, so it seems to be related to human development. A lot of times poor water quality can be a result of just poor agricultural practices, but when you get down to brass tacks and look at it, I don't even think there's more than 50 cows left on the island at this point, and they're in 2 fields. And 70% of the island is impaired. So I don't think livestock is a major issue, which can be a huge problem for upland, inland, and freshwater areas. And then if you even just the houses, there's only about 2,400 homes across the entire island, and that density, when you look at it, is about 1 house per 16 acres, which is very low density. Yet we still have this issue. And with how low the houses are, that means there's also generally not a whole lot of pets, there's not a whole lot of horses. There's really no smoking gun out there, except for septic systems. That is more than likely the issue, and that's the issue that we're trying to work on here at the Land Trust, and that's, we're trying to, through Brynne's work and Amy's work, and the whole team, trying to figure out to what degree that is contributing to these issues on Edisto Island, but it's fairly obvious and evident to us at the Land Trust that this is kind of the overriding thing. We don't see these issues in undeveloped areas where there's plenty of wildlife, but we are seeing it in more developed areas. It has to be something that humans are doing. And this is the obvious low-hanging fruit that we can try and work on. Septic systems are a necessary evil. They're not really evil, but they are absolutely necessary. The entirety of Edisto Island relies on septic systems, and only 40% of the beach is currently on municipal sewer. 60% of the structures on the beach are currently reliant on septic systems still. So this is something that impacts the entire community. When a septic system works, when it's properly sited, when it's in full working condition, it is not contributing any pollution to the watershed. However, if it's damaged, if it somehow in some way, shape failed, if it doesn't have a bottom onto the septic tank, or if it's just sited poorly, or local conditions have changed that now mean that the septic system is no longer functioning how it was when it was permitted and installed, that can result in raw wastewater being discharged directly into surface waters in our tidal creeks, which is not a good thing. And when you have a large percentage of septic systems that fail within a community, then that very quickly can become an island-wide problem with just a small percentage of failed septic systems. So the easiest way to try and get a handle on this is with septic system maintenance. We really don't think about our septic systems until they start acting up. When they fail, when you happen to have guests over and you've got raw wastewater spilling into the yard, we have all been there. That's generally the only time that a lot of us think about our septic systems. And a failed septic system is not only bad for the ecosystem, bad for the waterways, but it's also bad for your wallet, because that means you're going to have to get the thing repaired. So a little bit of preventative maintenance can go a long way. And I'm not pointing any fingers in this regard. My septic system failed catastrophically in 2020. It was 65 years old. We hadn't looked at it in 15 years. So this is an issue that can impact everybody. And we all need to be proactive with septic system inspection and maintenance to make sure that we are not part of the problem. And it's something, a septic system can begin failing years or decades before it actually totally fails in a way that becomes obvious to us and unignorable. And so doing regular maintenance and regular inspection on septic systems, make sure that we are not chronically and secretly accidentally polluting the waterways without our knowledge. And some other ways that you can help with water quality, each of us as individuals is picking up after our pets near waterways, reducing fertilizer use along creeks. You have waterfront property, maintaining a waterfront buffer, not mowing all the way up to the creek edge or all the way up to the marsh edge can really help sequester a lot of bacteria and nutrients on the upland before it reaches the creeks. And then just in general, take better care of your septic system. Because we're all responsible for water quality, and our actions and our inactions affect everyone within the watershed, everyone who relies on the waterways and creeks. So the big thing that we're doing here at the Land Trust to try and reverse our water quality issues here is the Edisto Island Septic System Improvement Program. This is a program that we piloted back in 2022, where we offered free septic system inspections and repairs to low-income landowners around Edisto Island. We did this with about a $24,000 grant from the Coastal Community Foundation. We inspected 15 systems. 10 of those needed repairs, and one of them needed to be fully replaced. But what that showed us is that septic systems absolutely are part of the problem here on Edisto Island. So with that, we were able to, with the watershed-based plan now being published, we were able to then seek EPA Section 319 funding to then go out and try and do a much larger program, as well as cover some education outreach programs. So any of y'all who came to those events over the last couple of years, thank you immensely. But with that, we were able to do a second wave of repairs. So we looked at 50 systems that we selected through the program, and we were absolutely flabbergasted by the fact that we had to repair 45 of them. 23 had to be completely replaced from outfall of the house to the end of the drain field. That's over 50% of the systems that needed repairs needed complete replacement. What is that? That is 90% of the systems that we looked at were damaged in some way, shape, or form.
Cost parameter of that. What was the cost parameter of
You're making me do math on the spot, Ted. I think it was about $200,000 we spent in repairs on that. My math is adding up, whatever. Whatever 4,900 is times 45. So, with that, we still had a ton of folks still on the waiting list for the septic program, and so I'm happy to announce that we have been approved again for a second EPA 319 grant, and so the program is coming back in 2026. We have hoping to have about $250,000 in funding to be able to repair at least another 45 damaged systems on Edisto Island. We've already got, I think, over 30 applicants on the waiting list to be processed and hopefully selected and get systems repaired. But we're hoping to incorporate new data, new research into the program to make it more efficient, make it more targeted, and have a much greater impact. And then additionally, we want to really kind of streamline the program to make it more efficient, try and get a lot of this work done as fast as we can. This is a 2.5-year program. I fully expect to have all the money spent again within a year like I did last time. But just know that the septic program is coming back, and if you know of anyone who needs help with septic system repairs, please give them my info, and I can get them hooked up with an application, either online or paper. And if you want to know about this, you happen to be in just the right place, because we have all the experts gathered right here in the room. Additionally, I have a lot of info on our website at edisto.org/water quality, and there's my contact info if you want to holler at me. Our office is across the road. Just show up there, and I won't be able to escape. That's all I've got. Thank you all for coming out. And we'll take questions.
So you've repaired about 75 septic systems so far?
We've looked at 75. We have repaired 55.
Okay. In the last year. So all of this was going on at the same time we're doing our sampling, so.
I fixed all of the problems, and so that's why
Yeah.
I'm
Anyway, it's just food for thought that,
Yeah.
some of, we were seeing lower bacteria levels at the same time that these improvements are happening in the watershed too. So
And that's
we don't have direct cause and effect, but there's definitely a correlation. There could be a correlation there.
And the one thing I want to highlight about this map that I have up here is that this is what is produced by the Shellfish Monitoring Program that DNR runs, which utilizes data produced by the South Carolina Department of Environmental Services. And this runs on a 2-year lag time. So this is the brand new data for 2025, which only includes the data collected up through the end of 2023. So none of the actual repair work that we did in 2024 is captured in this, and we won't know until the end of 2026 whether or not there has been a significant decline in these levels. Yes.
I have a question. The dark red area on the south Edisto, that doesn't seem like it computes with where people are. So why that area?
That has stumped us as well. My best guess on that is this area of Edisto Island fluctuates heavily and rapidly between brackish and saltwater. And so I think it's closed not necessarily for high bacteria levels, although there can be really high levels coming down the South Edisto after flood events. I think it's more so because of the health of the oysters. They may not, you have oysters that are, potentially dying from excessive freshwater or brackish events. And so people going out there and harvesting them, they might get sick, not because of the bacteria in the waterway, but because they're eating half-dead oysters. That's my best guess at the moment, but I haven't really been able to get a really firm answer on that.
Have you sampled the water coming down the south Edisto, say around the Jacksonboro Bridge?
We have not. Since we're mainly focused on the Edisto Island watershed and mainly on how it impacts oysters, we haven't, just because that's up above the saltwater.
So I can add something to that. Yeah. So when we got started with the watershed plan, what I was hearing from folks was, and understandably, we always say, we all live downstream, but y'all are downstream, right? You're at the end of the Edisto River. So we were concerned that what if some of the bacteria pollution is not coming from within the watershed. So we did look upstream at the impaired list, the list of impaired sites, and there's a long reach of the Edisto River above the Edisto Island watershed and up that does not have bacteria impairments. So that suggested to us that a lot of the bacteria was coming from within the watershed.
I hope I don't interrupt your time, but can I ask a question to the audience?
Absolutely. You can come up and take the stand if you want.
I'm not gonna get that professional. But as an outreach component of it in education, I'm always looking for different ways to spin the topics, get the information out to the public. So I really want to hear any wins or any light bulbs that went off into your head when some of this information was given to you at any point in time. Would anybody would like to share something that was like, I figured that out now, now that it has been presented to me. Some people, to give you an example, did not realize how much their garbage disposal was connected to their septic. And that was one of those aha moments for some homeowners. But is there anyone here that would like to share like something that they learned through all of this that is very impactful of making a change? Yes.
We just built a house and we had to go through a pretty rigorous process to get a septic permit. We had to do an engineered system, and that was very educational for me because I didn't understand why, you know, and it was pretty expensive too, but I learned a lot from the permitting process.
Awesome. And what she means by engineer is an alternative type of system, also called, is it anaerobic?
Yes.
I believe it is. So it's a system that is a little bit more technical. It's not like a gravity fed. It has pumps involved in that. You got some extra tanks. You got to put some disinfectant things in there. So she's able to have a hardier system than what your typical system would come out to be. Anyone else like to share?
Question on, how often do they recommend that septic tanks get inspected, just a normal beach house?
Oh yeah, so a normal beach house, if you are going within the specs of your home, so what I mean by the specs of your home is the individuals that live in your home is spec'd for the home. It could be 4, if you lived in Charleston with college students, it could be 9. So, but if you're going within that range, it's 1 to 2 years. The more pressure you add to it, the more you're gonna need to get it inspected because there's more influences going on with that system.
So inspected 1 to 2 years, pumped out, they recommend every 3 to 5 years. So the inspection is just to make sure everything's working. You know, when Tom went out and they were like, oh, the drain field's not even connected to, you know, the pipe from the house not even connected, that was a way to catch something that was causing pollution even if the tank itself wasn't full. So getting it inspected, and you know, we now like to call them checkups because inspection does, I've gotten feedback that sounds a little bit like regulatory, that you might come in and if something's wrong you might get punished. Like, you won't. It's just a good idea to have your tank checked out, just like you go to the doctor to get checked out. It might not need any work, but you'll know that it's functioning properly. But then on average every 3 to 5 years is when you want to have it pumped out, unless, like you said, you're kind of overloading your system. If you have a 3-bedroom house, it's probably, your septic tank size is probably spec'd for what, like four people in that residence. But if you're renting it, you know, or you have, you know, maybe your kids came back and they're living with you and the grandkids, and now you have more people in that house, the tank is gonna have to be pumped out more regularly.
Tom, I'm sure there's a lot of parameters, but is there a life expectancy, quote, of septic tanks and fields? I mean, you alluded to yours for 65 years.
A tank can last a very, very long time, especially the modern tanks, so long as there's no manufacturing defects. We have seen a couple of septic tanks on the island that, for whatever reason, use inferior cement on the T's inside the tank that had to have some, you know, touch-up repair work. But as long as you're not driving dump trucks over the septic tank all the time, the tank itself should last more or less indefinitely. But the drain field itself can vary highly. The more you use the system,
Of the ones you've repaired, what is the average age of those? Do you have any idea?
They range from 70 to 5.
70 to 5 years?
Yeah. So the 5-year-old ones have been misused or you know, if you flush a lot of stuff down the tank that isn't compatible with the drain field, or if you overuse the tank, then stuff can very quickly get into the drain field, clog the porous pipe, and you have to get the drain field replaced. Generally, most of the tanks that we have replaced are just like homemade cinder block tanks that were built back in the 60s that didn't have a bottom, or they're just literally starting to cave in.
Cinder block.
But
There is kind of a rule of thumb that the lifespan of a septic tank is around 30 years. However, you know, I have been out with septic contractors who are pumping them out who will say that, you know, they'll see a 50-year-old tank that's solid as the day it was installed, and you can have a new tank that has tree roots growing in through the pipes into the tank or puncturing the wall. So age, you know, I when we were doing the watershed plan, Charleston County did have mapped septic tanks for the island. We found out that they probably only had about half as many as are on the island, but they also had the age of install. And we were just assuming, oh, these older tanks are gonna be the ones that are causing all the problems, but sometimes what we see anecdotally is that the older tanks on older houses were put in, those houses are probably on the high ground, right? Like the soil conditions are actually maybe better for that tank to function well. In some of the newer houses, if you're not putting in an advanced engineering system, you might be putting in a brand new septic tank, but the drain field's just not gonna work well because the soil conditions aren't great for septic. So that age thing is something that I've kind of shifted on a little bit. I don't necessarily worry as much as if it's old, as long as it's being regularly inspected. And sometimes the new ones, I'm like, well, should they have even gone in there? That site, you know, doesn't drain. So
yeah, site conditions play a huge role in kind of how much latitude a system has where it will still be functional. So a really high elevated system on a really well drained soil, you know, might still be able to function even, you know, at a quarter of its capacity if there's only 1 or 2 people living in the house. Whereas a house with a lot of people in it on a really low elevation area with a really high water table, it might fail, you know, on a king tide if there's 1 too many people in the house. Kind of thing. And a system can also fail temporarily based off of environmental conditions, especially with drain fields. It's not necessarily a hard stop, but the system can stop functioning for a period of time if water tables get too high. And then once those water tables recede, start working again. But then that becomes a chronic pollution source, so that's part of what we're working with replacing systems. Is finding those systems that are like on the edge of failure and making sure they get relocated or improved in a way that they don't cause chronic issues.
So the cost for this, there's a cost to have an inspector come. So you go, who does the inspector? Is that a private company or is it the same people that do the pumping?
Depends.
Yeah.
It depends on their services.
There's sometimes there's more folks who can do inspections than people who can do pump outs, because pump outs you have to have the truck, whereas someone can be, you know, licensed and certified to do an inspection and not necessarily have the equipment to do a pump out. So it just depends person to person, but use someone who's certified by DHEC.
can we give a shout out to the septic guide?
Oh yeah, we Clemson Extension had a team together that's called the Be Septic Safe Team, and we created a guidebook. I don't have any with him because I'm
pile
always
at my
giving them
office.
out. Yeah, I'm sure he has some at the office, but in that guidebook, you are all our green people. We call green people who are interested, want to make a change, but if you have a neighbor or if you have someone who may not attend these groups or in your church, give them that guidebook to give them something that they can look into and fold into to find out how they can improve their septic systems. A lot of times I grew up on septic systems with my father, and he's upstate. I'm from Greenwood, South Carolina, and he was very militant about that, and we had no water near us to be concerned about. So with him having that type of mentality gave me the same mentality. Got a job now, but gave me the same mentality to share that information the same way too. So use those guidebooks. If you need any more time, I got plenty in the office and we can print more. Even if you have a business that's connected to it, we can add a logo that's with it. All of those things can be done. And I also have like little trinkets and tools that help people maintain their septics in a better way to get those good best practices.
I'm following Tara's advice, like how does this affect me? I live on the beach, and this is a small thing, but they put the animal bags and the waste bags right up on the beach next to the walkway, the accesses to the beach. There's no, and so that seems like that would help, you know, people get the bag and all that. Well, they think sometimes that they can just put the bag right back where they got it with the full of poop. They just expect somebody to pick it up. Or, you know, it happens rather often. And I just read something like if there's a waste can nearby, maybe that would solve the problem. But that seems like a kind of an overkill. There is a garbage can at the main when you get back to the main entrance.
We did put that in the watershed plan actually as a recommendation, is to have garbage cans where you have the
wow.
waste bags.
And that somebody would regularly pick up. Yeah.
Well, and that's the problem, is that probably has to be through the town, I guess, or
Yeah.
whoever
They have to
does
regulate
the.
Yeah, in James
Yeah.
Island, their stormwater project manager had a task force that was set up with putting, they said, they're doing the right practice, they're putting it in the bag, but when it gets to the bag, it stops there. It either ends up in leaf litter, it ends up everywhere. You
Just
kinda
thrown
have
in the
to,
river still.
yeah, you kinda have to tote someone along the way. So what they did with their town and the stormwater planner, they put just trash cans around them. Now, they did have to collect those trash cans and make sure they're empty, but they did not end up back into the waterway, or what's worse to me is putting it in leaf litter that ends up in compost. So I think it takes a little and it gives a little, you know, you have to go that approach. But if you are trying to be that go-getter and you put that trash bin out there, have one with a lid, you know, have your gloves in your car so you're not having to touch it. Make it as an easiest process for yourself as possible. That's what I do. I keep everything in. Anybody can ask me for anything in my car, and I have it. Zip ties, all kind of stuff. But I'm just that water advocate like that. But if that is something that you're noticing, sometimes someone seeing your behavior will change their behavior.
Well, and then there's also the times we get flooded, and if there's a trash can out there, that's a big, but.
Yeah, and that too, but you can put it higher and have a sign that's directing trash goes here. You know, it's just to make it a little bit easier on you. And signs go a long way, even though a lot of people don't read, some people do.
Any other questions? Thanks to all these
Thank you.
folks. It is wonderful and